I'm Vishnupriya Kasireddy, and I'm a biology graduate student at Marshall University in Huntington, West Virginia. I'm modeling the relationship between host fish availability and the reproduction success of freshwater mussels. Mussels are bivalve mollusks in the order Unionida that live in freshwater systems. They're the second most diverse group of organisms in terms of described species, reaching their highest diversity in North America east of the Mississippi River. Research in freshwater mussels is growing for two main reasons. First, they're among the most endangered fauna on Earth. Nearly 70% of North American species are already considered extinct or imperiled due to water pollution and changes in sediment composition from dams. Second, they have a remarkable life history and play an essential role in aquatic environments by continuously filtering out algae, bacteria, and other organic and inorganic matter from the water columns. Declines in mussel communities are attributed to human-related impacts such as climate change, acid deposition, and development of dams and bridges. As you can see, the changes of the Ohio River in the 1890s and the river today. These impacts increased water depth and altered the natural seasonal variations in temperature, oxygen regimes, and changed how sediments are transported and deposited. The Ohio River is one of the most diverse and essential ecosystems for mussels, providing habitat for up to 80 species. So how do mussels reproduce? Freshwater mussels have a unique and complex way of reproduction, requiring a parasitic relationship between each mussel species and a fish host to complete the cycle. Some mussel species require a specific host, while others are a generalist species with host variability. The life cycle of a freshwater mussel begins when a male releases sperm into the water column, which enters the female through her in-current siphon. It is believed that temperature and photoperiod trigger this release of sperm. The eggs are then fertilized within the marsupial, and then a lure, which is an extension of the mantle as shown, is produced. A passing fish, acting as a parasitic host, is attracted and bites the lure. Upon attack, the young mussels, known as Glaucidia, are then released into the fish's mouth and attach themselves to the gills, fins, and skin. After feeding on the fish during their larval stage, they detach themselves, settle onto the bottom, and begin their adult stage. Now that we know how mussels work, let's go into the methodology. The survey sites as a green-up pool are mapped below, where each site is characterized as fixed station, random, or revisit. 6 100-meter transects are placed at 100-meter intervals perpendicular to flow using a lead line at each site. Each 100-meter transect is then divided into 10-meter intervals. Surveys are conducted on the upstream side of the transect that is 1 meter wide for surface mussels and juveniles using scuba. So what is the probability that a fish would come in contact with a mussel? To better understand the model, let's look at a small-scale survey site below. The circles represent mussels and the triangles represent host fish. We assume that a fish will come in contact with a mussel if it is within a 1 meter radius. So the area that m number of mussels occupy is m times pi r squared over the length times width of the site, or the site's area. Raising this quantity to the number of fish hosts available over the total fish available gives us the probability of an host fish coming in contact with a mussel. Assuming everything is constant, as the number of mussels and the availability of fish hosts increase, the probability of reproductive success approaches 1. Going further, my study will determine if, how often, and under what circumstances mussel populations are limited by the availability of hosts. Thank you.